Alright, so you learned all about genetics now in Punnett squares and how we pass on genes, how proteins are made, and everything about like genes and how genes control our body. Now we're going to learn about what we can do with these genes. So scientists have found ways to manipulate these genes, and it's called genetic engineering. And we're going to talk about several different genetic engineering. And the first one we're going to talk about is the case of the glowing kitten. So, with genetic engineering, we can actually make this cat here, this is real, glow in the dark. All right, this jellyfish is natural and naturally glows in the dark, so it's born this way. Whereas this cat is not born this way. Cats don't glow in the dark. We made it glow in the dark by doing something to it. So what you need to do is just, these are just predictions, so whatever you write is fine, but just think about it. Think about how this one glows naturally, and then think about how we could have made this cat glow in the dark. Alright, then what you're going to do is you're going to pull up, there's a PowerPoint on the website, and it looks kind of like this. Alright, so here's one all about the glowfish. So the glowfish are fish that is a zebrafish, but we actually made them so they glow, just kind of like that cat or those mice. So what we did to make the glowfish is that we inserted a gene from a jellyfish into the zebrafish to make it glow. So we took a gene from this jellyfish that glows and put it into the zebrafish so it glows in the dark. What we did, why we did it, which is to show the level of pollution in the river. So these guys die very quickly if the river is heavily polluted. So the more of these that you would see, you would think that the river is going to be cleaner. And then we can actually also sell them as a pet. So what you're going to do on your thing here is you go down to glowfish, which looks like I didn't even put that one on here. So never mind, you can skip that one. But what you would have done is I would have said, okay, this is the zebrafish. That's the organism involved. The gene is from a jellyfish. What's the point? It's we made them colored so we can identify how much pollution is going on in the water. And then there's a question on the poster. And this is, how does this fish benefit us? Well, it looks cool. And it also helps us see levels of pollution in the river. Then this is the super spuds. So the super spuds or the potato, that's where you're going to go to here, the potato here, and then you'll put what the organism is, where the gene came from, and so on and so forth. And you just fill this out. So these are other organisms that have been genetically modified to do something that it doesn't normally do. All right. So once you're done with that, then you're going to look at, okay, how do we genetically modify things? So we're going to talk about something called gene splicing. So gene splicing, you're going to go through and read these, and you should be able to read this stuff and get the answers right from the reading, as long as you read it carefully. And when you get down here, get the sheet of paper that has the cat and jellyfish gene DNA on it. So what you're going to do is look at the cat DNA here, and here's our jellyfish DNA. So the first thing you're going to do is you're just going to focus on the jellyfish chromosome. So you're going to cut out these 4 strips of the jellyfish DNA. So you're going to cut out these 4 strips, and then you're going to, you see how this symbol is the same as this one? So you're going to tape or paste these two together. This symbol matches this symbol, so you're going to tape or paste these two together. And that symbol matches that symbol, so you're going to tape or paste that one. So you have a strip, one single line strip of this. And then you're going to find this sequence, which is the GFP gene. So it tells you what the GFP gene is up here, but it's the gene that makes the jellyfish glow. It produces a green fluorescent protein. So you're going to highlight that one on there. And then you're going to leave that alone for a second, and then you're going to take the cat DNA here, and you're going to cut these out and again paste these together. Now this one you're going to make it into a circle. So this one gets next to this one, and then this part connects to that part, so you'll have a circular piece of DNA. Alright, then what you're going to do is we're going to cut along these dotted lines here. So there's 1 on the cat DNA, and there's 2 on the jellyfish DNA. And then you're going to paste the jellyfish DNA into the ring of the cat and connect it together so it's another ring again. And then what you'll see is that means we actually cut out the jellyfish DNA and put it in to the cat's DNA. So now the cat has this GFP gene, so now it can make the protein. Alright, so I basically just gave you the answer for this. So then what you want to do is just summarize that whole process that we just did. What did we do? What did we started with a gene, so what was the gene that we started with? What did we do to that gene? Um, and then how did we make the cat glow? All right, and then you'll just go back to the original problem and say, why is this cat glowing? So you should have figured that out by now after through that stuff. And that is gene splicing. That's the biggest thing. So splice means to cut, and that means gene. So if that doesn't give you anything, cut genes, that's pretty obvious. All right, so, and you can ask me more questions if you need to.